Welcome to G Fun Facts Online. Today, we are pushing past the ultimate speed limit of biological history. For over a century, the study of evolution has been a journey backward in time. We trace our lineage from primates to early mammals, back through the murky waters of the Devonian fish, past the Cambrian explosion, and down into the microscopic realm of single-celled organisms. Eventually, all lines converge at a singular point known as LUCA, the Last Universal Common Ancestor. Living roughly 4.2 billion years ago, this organism, or population of organisms, was the genetic bottleneck through which all life on Earth passed. Every bacterium, every fungus, every plant, and every animal today carries the molecular signature of LUCA. For decades, scientists believed that peering beyond LUCA was impossible. We called it the biological event horizon. We assumed that the pre-LUCA world was a chaotic, primitive scramble of chemical evolution that left no trace in modern genomes. But we were wrong. A revolutionary shift in evolutionary genomics has shattered this event horizon. Recent breakthroughs, culminating in landmark research published in early 2026 by a team including Aaron Goldman, Greg Fournier, and Batul Kakar, have unveiled a method to see through LUCA. The key to this time travel lies in a specific, rare, and powerful class of genetic artifacts known as Universal Paralogs. This is the story of pre-ancestral gene duplication, the mechanism that built the complexity of life before life as we know it even began. To understand this pre-ancestral world, we must first understand the engine of complexity, which is gene duplication. In 1970, the geneticist Susumu Ono published a seminal book titled Evolution by Gene Duplication. He famously argued that natural selection merely modified, while redundancy created. His insight was profound. If a gene is essential for survival, natural selection forbids it from changing too much. A mutation that breaks a critical enzyme kills the organism, meaning evolution is stuck in a conservative loop. But if that gene accidentally duplicates, if a copying error results in two identical versions of the gene sitting side by side in the genome, the shackles are loosened. One copy can continue doing the essential work, maintaining the status quo. The second copy, however, is redundant. It is free from the intense pressure of survival. It can mutate, drift, and explore new chemical possibilities. Over millions of years, this spare tire can acquire a new function or split the workload with its twin. The new frontier of science asks a daring question. Did gene duplication happen before LUCA? If a gene duplicated after the domains of life split, we call the copies paralogs. But if a gene duplicated before the last universal common ancestor, then both copies would be inherited by LUCA, and subsequently by all of LUCA's descendants. These are the universal paralogs. They are the fossils of the pre-LUCA world. Because every modern organism, from E. coli to Albert Einstein, possesses both copies of these ancient genes, we know that the duplication event must have occurred in the deep, dark past, prior to the divergence of bacteria and archaea. By comparing the sequence of copy A against copy B across the entire tree of life, we can triangulate their point of origin and mathematically reconstruct the sequence of the ancestral gene that existed before the duplication. Let's look at one of the most spectacular examples of pre-ancestral gene duplication found in the machinery of bioenergetics, the ATP synthase. This is the enzyme that powers the world. Sitting in the membranes of our mitochondria and the cell membranes of bacteria, ATP synthase acts like a rotary motor. It uses the flow of protons to spin a turbine, crushing ADP and phosphate together to make ATP, the universal energy currency of life. For years, debates raged about whether LUCA could do this. Was LUCA a primitive scavenger feeding on chemical soup, or was it a sophisticated entity? The answer lies in the universal paralogs of the ATP synthase complex. The motor consists of a rotating stalk and a catalytic head. The catalytic head is made of alternating subunits, often called alpha and beta in mitochondria. Structural analysis reveals that the alpha and beta subunits are remarkably similar in their 3D shape. They are clearly cousins, descended from a single ancestral protein. Crucially, both bacteria and archaea have ATP syntheses composed of these two distinct subunits. This implies that the duplication event, the moment a single proto-subunit gene copied itself and diverged into the alpha and beta forms, happened pre-LUCA. This discovery rewrites the biography of our oldest ancestor. It means that the pre-LUCA lineage had already evolved a complex, multi-subunit rotary nanomotor. Before the duplication, there was likely a homo hexamer, a ring made of 6 identical proteins. This primitive machine probably worked, but inefficiently. Then, the gene encoding this subunit duplicated. One copy became the specialized catalyst, the hammer that forges ATP. The other copy lost its catalytic ability, but evolved to become a structural scaffold, a regulatory wall that improved the stability and efficiency of the ring. By the time LUCA appeared, this heterohexamer was already fully formed. LUCA was not a half-alive blob, It was an energetic powerhouse, equipped with a Ferrari of molecular motors. Now, let's look at the blueprint itself. How did life learn to read DNA? The translation of genetic information into protein requires a translator, a molecule that knows which amino acid corresponds to which triplet of DNA bases. These translators are the amino acid tRNA synthetases. There is one enzyme for each amino acid. Tracing the lineage of these enzymes reveals one of the most profound chapters of pre-ancestral history. Researchers analyzing the class 1 enzymes discovered that the enzymes for tyrosine and tryptophan are universal paralogs. They look like siblings. This suggests that deep in the pre-LUCA past, there was a single enzyme, a generalist, that likely recognized a broad range of bulky aromatic amino acids. It was not picky. It might have grabbed either tyrosine or tryptophan, or perhaps a rudimentary ancestor of both. Then came the duplication. One copy refined its grip on tyrosine, the other copy refined its grip on tryptophan. This is pre-ancestral gene duplication in its most potent form, the expansion of the genetic code itself. The genetic code did not appear fully formed, it grew. The pre-LUCA code was likely fuzzy, where fewer amino acids were used, and the machinery couldn't distinguish between similar ones. As the genes duplicated, they allowed the code to become more granular. The universe of chemical building blocks expanded. The addition of tryptophan, often considered one of the last amino acids added to the standard repertoire, was enabled by this specific pre-LUCA duplication event. This tells us that the dark age before LUCA was the period where the dictionary of life was written. To build a protein, the ribosome also needs a delivery service. Molecules called elongation factors shuttle the amino acid-loaded tRNAs into the ribosome's active site. In every domain of life, we find two distinct types of these factors. In bacteria, they are called EF2 and EFG. In eukaryotes like us, they have different names but perform the same functions. These two families are universal paralogs. They are structurally nearly identical, utilizing GTP energy to change shape and push molecules around the ribosome. The duplication that created these two factors is one of the oldest known events in biological history. We started with an ancestor, a primordial GTP ACE that likely helped the primitive ribosome move or bind tRNA loosely. Then the split happened. The gene duplicated. Copy 1, specialized in delivery, carrying the ingredient to the chef. Copy 2, specialized in translocation, moving the conveyor belt of the ribosome after the bond is made. This separation of duties was a massive leap in efficiency. It transformed protein synthesis from a stuttering, error-prone process into a high-speed assembly line. Because both bacteria and archaea have this dual system, the high-speed assembly line must have been completed before LUCA. This challenges the progenote hypothesis, which is the idea that LUCA had a primitive, sloppy translation system. The evidence from Universal Paralogs suggests otherwise. LUCA was a sophisticated cellular machine with optimized protein synthesis. But how do scientists actually do this? How do you reconstruct a gene that hasn't existed for 4 billion years? The methodology is a triumph of computational biology, blending phylogenetics with ancestral sequence reconstruction. Constructing the tree of life is difficult because the roots are deep and muddy. Bacterial and archaeal genes have diverged so much that comparing them is remarkably difficult. However, universal paralogs provide a cross-brace. Because gene A and gene B exist in the same genome, they share the same history after the duplication. But because they diverged before the organismal lineage split, they provide two independent views of the same history. By forcing the species tree implied by gene A to match the species tree implied by gene B, scientists can error-correct the deep phylogeny. It is like having two independent eyewitnesses to a crime. Where their stories overlap, you find the truth. Furthermore, universal paralogs help with reciprocal rooting. Finding the root of the tree of life is notoriously hard because standard trees show relationships but not direction. Universal paralogs solve this. You can use the paralog A tree to root the paralog B tree, and vice versa. This technique confirmed the fundamental split of life into two primary domains, bacteria and archaea, and placed LUCA firmly at the node connecting them. Once the tree is built, researchers use maximum likelihood statistical models to infer the most probable amino acid sequence of the ancestral nodes. They project back to LUCA, and then they project back further to the ancestor of the paralogs. Labs can then synthesize this DNA, inject it into modern bacteria, and produce the ghost protein. When tested in the test tube, these resurrected pre-LUCA proteins are often extremely stable and promiscuous, capable of multiple functions, which supports the idea of early generalist enzymes. The study of pre-ancestral gene duplication has revealed a lost epoch of Earth's history. We can now conceptually divide early evolution into three distinct eras. First, the origins, or abiogenesis, which was the rise of simple self-replicating molecules. Second, the Dark Age, or the pre-LUCA epoch. This is the newfound gap between the first cell and LUCA. It was a time of rampant gene duplication and high evolvability, where the complexity of the cell was assembled piece by piece. And third, the extant age, from LUCA to today. This dark age is vital because it solves a paradox. If LUCA was 4.2 billion years ago, and Earth became habitable perhaps 4.3 or 4.4 billion years ago, that leaves a very short window for life to evolve from nothing to a complex cell. The sheer number of universal paralogs regarding membranes, transport, coding, and energy implies that the pre-LUCA epoch was a pressure cooker of evolution. Evolution may have moved faster then than it does now, without the rigid error correction mechanisms of modern cells, genomes duplicated, fused, and recombined wildly. It was a golden age of duplication. While we focus on the deep past, it is vital to recognize that this mechanism didn't stop with LUCA. The pre-ancestral theme echoes through time. The most famous analog is the 2R hypothesis in vertebrates. Just as pre-LUCA duplications created the cellular toolkit, the pre-vertebrate duplications created the anatomical toolkit. For example, invertebrates have one cluster of Hox genes, while vertebrates have 4. This quadrupling allowed vertebrates to build complex skeletons, jaws, and brains. The difference is the timescale. The vertebrate duplications happened around 500 million years ago, while the universal paralogs happened over 4 billion years ago. But both followed the same logic. Duplication breeds freedom, and freedom breeds complexity. The discovery of universal paralogs is just beginning. As artificial intelligence improves our ability to predict protein structures, we can recognize paralogs that have diverged so much in sequence that they look unrelated to the naked eye. We may find that many more of our genes are universal paralogs than we currently realize. Perhaps the enzymes of glycolysis or the components of the cytoskeleton hide ancient duplications that predate the cellular era. The ultimate question remains: if we can reconstruct the duplication events before LUCA, can we reconstruct the first duplication? Can we find the original gene from which all protein-based life descended? Probably not. The signal fades into the noise of deep time. But we are getting closer. We are pushing the event horizon back. We now know that before the tree of life sprouted its main trunk at LUCA, there was a root system that was tangled, complex, and driven by the creative force of duplication. We are not just descendants of LUCA. We are the inheritors of the twins born in the dark waters of the Hadean Earth. The redundancy in our genomes is the legacy of that ancient struggle to survive, where 2 genes were better than 1, and a copying error became the spark of civilization. Pre-ancestral gene duplication is more than a molecular mechanism. It is the geological strata of biological information. It tells us that complexity was not a late addition to the story of life. It was baked in from the very start, forged in the pre-LUCA fires by the duplication of essential machinery. When you take a breath, thank the pre-ancestral duplication of globin genes. When your cells burn food, thank the pre-ancestral duplication of ATP synthase. When your body builds muscle, thank the pre-ancestral duplication of elongation factors. We are, in every sense, a collection of ancient echoes, amplified and refined over 4 billion years, but still singing the same song that began in the dark. Thank you for listening to G Fun Facts Online.